So I'm going to be talking about work I've done both as a PhD student with Susan Harrison and now currently as a postdoc with Carla D'Antonia and Peter Adler. So I'm particularly fond of California grasslands for a number of reasons. They're easy systems to work in as an ecologist. They're pretty. They have a lot of really cool species. So getting into it here. Let me just start by acknowledging my collaborators. I already mentioned Susan Harrison and my PhD lab, but also Carla D'Antonia and Peter Adler. So grasslands occur across a large extent of California, and they occur in a lot of climatic regions, including high rainfall regions and drought regions. And so you can have coastal prairies, which get a fair amount of rainfall, both in the winter through rain and then in the summer through fog. And you can have drier inland grasslands. This is from Sedgwick Reserve, which gets very little precipitation in many years. They also occur in a number of soil types. So here is a non-serpentine grassland from McLaughlin Reserve in Lake County, and then a serpentine grassland that occurs not very far down the road from this one. And I think this is what makes these grasslands so interesting for me. So you can see in this next slide, actually, how different serpentine and non-serpentine grasslands can really be in species composition. So you can have non-serpentine grasslands that are predominantly dominated by non-native annual grasses. Maybe you have a few dominant native forbs. In other cases, to find a native forb in there would be really challenging. In contrast, serpentine grasslands tend to be these open landscapes dominated by a lot of short-statured native animals. Someone mentioned earlier how much she loved the tiny little plants. It's part of why I love serpentine. And in some cases, they run right into each other. So you have serpentine grassland in the foreground, and you can literally walk across that ravine into non-serpentine grassland. And, I mean, I think that's a fairly dramatic picture of the differences. You might expect that because of these differences, they might respond to climate differently. And that's kind of the question that motivated my work. So here's just a breakdown. For those of you not familiar with serpentine soil, which is probably very few here, it's really low in nutrient availability. It's low in nitrogen, phosphorus, and especially calcium. It's high in magnesium, so it has this characteristically low calcium to magnesium ratio. And they're dominated by annual forests, as you've seen. Non-serpentine, in contrast, is relatively fertile, highly invaded by a lot of avena, bromus, and other species like that. And this table sort of points out some of the differences in the functional groups. So in terms of species richness and cover, serpentine communities have more annual forbs. And they also tend to have more perennial forbs than grasses. But like I said, the big difference between these two grasslands is the amount of non-native grass cover, which is pretty dramatic. And these are averages of about 40 non-serpentine and 40 serpentine sites. So in some cases, this figure here can be 100%. So basically, to sum up my work, I've been motivated by how does soil type influence community response to climate. And over the years, I've taken different approaches. I'll talk about some experimental work I did as a PhD student, and then some long-term observational data that I analyzed. And I'll conclude with sort of a brief description of what I'm doing for my postdoc, which is taking a two-pronged approach to understand mechanisms. So first, in the first study, I basically manipulated rainfall in a serpentine and non-serpentine grassland. So I set up drought shelters, which reduced rainfall by 50%, or I had these irrigation tanks that increased water about 30 to 50%. And then I monitored the biomass production of the community. There was also a component of looking at serpentine endemics, but I don't have time to talk about that today. So after 3 years of this, I found that increasing rainfall on non-serpentine soil led to an increase in community biomass. Not really that surprising. But there was no effect of altered precipitation on serpentine at all. It's like a flat line over there. So it suggests that, at least in terms of productivity, these stressful communities might be more resistant to climate change than others. But we still don't really know what that means in terms of species composition or their long-term dynamics, which is where the next section comes in. So in this study, I was using Susan Harrison's data over these 80 grassland sites at McLaughlin Reserve. There's 42 that are on non-serpentine and 40 that are on serpentine. And every year for, I think it's up to 16 years now, but at the time of the study it was 10, the lab has gone out and measured community composition and abundance. And so I took those data and I analyzed them by measuring how much turnover over time was, and I'll explain that. So what I mean by turnover is change in species composition from one year to the next, not just in the identity of the species, but in their relative abundance. So let's take two imaginary plots. Both have 4 species at some relative abundance, and let's say they start out identical. The expectation that we had for non-serpentine soils was that there would be large changes in the relative abundance and species richness. So in this case, all of these 3 species have shifted abundance. This one's dominant now, and we've lost that little rare magenta species. However, for serpentine, based on our previous study, we expected very little change. Maybe you lose the rare species, but the other species maintain their relative abundance. So I calculated a measure of this turnover called Bray-Curtis dissimilarity, and it basically asks if the same species were in the plot in the second year as they were in the first year, and if they were there in the same relative abundance. So what you need to know here is that low values on this axis indicate little change in composition, and high values indicate a lot of turnover. You can see this is not the result I was expecting. Serpentine communities appear to be less stable. But if you recall that big difference between the grasslands, which was non-native annual grasses, one might imagine that they could influence turnover patterns. And in fact, other researchers have found annual grasses can be highly dynamic on serpentine soils. So I took them out of the group, out of the data set, and I find that serpentine communities are more stable, so they change less over time in response to climate. But really the native community changes less over time. The annual grasses do change quite a bit on serpentine. So, we're talking about a particular functional group that's really different from a lot of our native flora. So maybe it's species traits that are driving community resistance. So we went out and measured a bunch of traits associated with stress tolerance. Those included height, specific leaf area, and leaf water content. Those are basically associated with photosynthetic capacity, growth rates, competitive ability. And our expectations were that non-serpentine communities should be tall, have big, broad leaves with high water content, and the opposite for serpentine. They should be pretty short, tough little plants. And so we collected all these traits for a bunch of species, and we calculated community mean-weighted traits, which basically takes an individual species traits, multiplies it by its relative abundance, and then sums it up over all the species so that you get a picture of what the community looks like. And we found that serpentine communities were indeed more stress tolerant. So here's 2 examples. So mean height was shorter on serpentine, as well as specific leaf area. We also needed to know if this correlated with turnover. And I'm not going to show you the figure, but I will tell you that when we relate this index of stress tolerance to that Bray-Curtis dissimilarity index, we find that it does predict it. So what we found was that plots that tended to be more stress tolerant changed less over time. And that was actually true on both serpentine and non-serpentine soil. So from this study, we learned these low fertility soils do seem to contribute to stability in some way, perhaps by selecting for species with stress-tolerant functional traits. And this stress tolerance contributes to stability. What I didn't mention in this part, because I didn't have time, is that we did find some evidence for a direct role of soil, but it wasn't soil fertility. So soil fertility did not predict stability. So it's still unknown what it is. And that's where my postdoc has come in. there was some work to suggest that microsite heterogeneity in some soils can actually buffer species from climate change because it gives them more niches that they can partition themselves out along, and so they don't compete as much. But that wasn't tested, so that's what I wanted to do. So to start to understand this, imagine you have a single species occurring in the absence of competition, and it takes up about this much space along a soil moisture axis. If we have a shift in rainfall from a mean here down, say as a result of drought, we see the species maybe drops in abundance, but it still exists. If we add the presence of competitors, everyone has a narrower realized niche, right? Because they're competing with one another. So now if we have that same shift in soil moisture, we lose both the blue and the red species. They're outcompeted by the black species. To get heterogeneity to play a role, we have to add another axis of variation. So in this case, we're just using pH as an example. So now each species has a much broader realized niche, and they can still exist if we get that same shift in drought. So this is the idea. So to test this, I set up a mesocosm study in which I grew species either in monoculture or in competition on 3 soil texture treatments, either clay, sand, or a mix of those. And I either allowed them to have ambient rainfall or I put a rainout shelter over them. And for the first 2 years of the study, I used Plantego erecta and Salvia palmari as focal species. And this is kind of what it looks like. So this is a subplot here. Each one contains 9 cells. For homogeneous clay, all 9 of those cells are clay. For homogeneous sand, all 9 are sand. For the heterogeneous subplot, I created a mixture of clay and sand. And then those subplots were nested within a larger plot that I applied the climate treatment to. And so that's kind of what the mesocosm looks like overall. Here's a picture of one of the plots from the ambient treatment and a picture of one of the plots from the drought treatment. So I measured seed production for all of these species, which was a lot of work, actually. Turns out salvia produces a lot of seed when grown in a mesocosm. And then we ran it through a simulation model, and I'm not describing the model because I think that might be kind of boring right now. So this is basically what we get. So we ran it through a simulation model to get multiple generations. And what we find is that salvia, which is the blue, just swamps Plantego. So not shocking, Plantego is going to go extinct, no matter what substrate it's on. But when we calculate the relative differences, we find that actually these heterogeneous subplots get swamped a little less. So it does suggest that heterogeneity can reduce the fitness difference between species, although it's not acting through competitive mechanisms. It could be acting through this thing called abiotic nonlinearity. So perhaps the interaction of the clay and the sand together, soil moisture doesn't dry out quite as much as if it were in sand or just clay. So that's basically what I just said. We think heterogeneity can slow exclusions. I'm still exploring this with some other species and other mechanisms, so I hope to talk about it again someday. But in general, what we find is that stressful, complex environments could buffer species from climate change or from communities, and there seems to be a role both for the direct and indirect effect of soil types, and some evidence to suggest the role of spatial heterogeneity. However, that said, Susan Harrison will speak later tonight and discuss that that conclusion may not be quite as 100% as we thought, that there's some other caveats. So that I will take any questions.
What's the mechanism of competition in those experiments? Because it looks like their roots are isolated from one another. Is that right?
So I should have mentioned, let me go back. I should have mentioned that each of those cells, for the competition plots, each cell was seeded with both species. Got it. So, and in the subplot, the roots can move around, but the subplots are separated from each other with waterproof barrier.
In serpentine grasslands, there tend to be a lot more perennial grasses, native perennial grasses. So between the serpentine and the annual grasslands, I didn't see that variable kind of pulled out. So I would expect less variation in the serpentine just based on the plants holding their spot, because there's a lot more perennial root systems, which tend to endure climate change and variable climate a lot better than annuals.
Yeah, absolutely, and we thought that could be a possibility as well, that just the presence of the perennials, but actually perennial abundance doesn't predict stability at all. So you're looking at 1 meter by 1 meter plots, so you get some perennials in there. It could be at a larger landscape scale, they contribute to stability, but at these smaller scales, they're just not common enough in these grasslands, we think, to play enough of a role. Again, if you have a higher abundance of perennials, I would absolutely expect that.
Hi. I was wondering if you have made any observations or studies in the field looking at sort of that experimental scale of heterogeneity, seeing if maybe that's what Susan's going to talk about tonight too, but seeing if that corresponds in terms of like the diversity or richness you see in these communities. Like, do you see more diversity on these heterogeneous soil?
I don't have any particular measurements of it, but if you've ever been out to a serpentine grassland, you look down and you can just see this microtopography, and there's differences in soil texture compared to these non-serpentine grasslands I worked in, which was like sandy or sandy loam. I'm actually scheming to set up a project this month to actually manipulate heterogeneity in resources by basically microdosing with nutrients in small patches and see if that
Very cool.
Yeah, thank you.
Have you done any studies on or done any monitoring on soil moisture, since the clay mineralogy is different between the serpentine and non-serpentine?
Yeah, and it's so variable. A lot of people like to think serpentine is this really amazingly droughty environment, but you get really clayey serpentines. I have one site where volumetric water content is much higher on serpentine than non-serpentine. So while it could be playing a factor within a particular grassland, I think that may not be like the big measure of variation between serpentine and non-serpentine. Yeah. It's an interesting question though. Yes.